Chapter 8.1, Identifying DNA as the Genetic Material. The key concept is that DNA was identified as the genetic material through a series of experiments. In this section, we're going to talk about several of those experiments. In the first experiment, in 1928, an English medical officer and geneticist named Frederick Griffith discovered what he called the transforming principle. Today we call it DNA, but at the time they had no concept of what that was. Griffith was trying to make a vaccine to prevent pneumonia infections in the Spanish influenza pandemic after World War I. The experiment began by using two strains of the Streptococcus pneumoniae bacterium, the S strain and the R strain. The cells of pneumococcus are usually surrounded by a gummy capsule made of a polysaccharide. When grown on the surface of a solid culture medium, the capsule causes colonies to have a glistening, smooth appearance. These cells are called S cells, the S for smooth. However, after a long time of cultivating, they noticed that some of those cells lost their ability to form that smooth capsule. Instead, the surface of their colonies was now rough and wrinkled. That's where we get the R form. With the loss of that capsule, the bacteria also lose their ability to infect healthy cells. Injection of a single S form of the pneumococcus bacteria into a mouse can kill the mouse in 24 hours or less. But we can inject over 100 million R cells and it's completely harmless. However, Griffith found that when living R cells, which should have been harmless, and dead S cells, which also should have been harmless, were injected together, the mouse became ill and the living S cells could be recovered from its body. This process was called transformation. As you can see here, they used heat to kill the bacteria, which we knew at the time was a valid way of killing off bacteria. Next, Oswald Avery and his colleagues, Colin MacLeod and Macklin McCarty, who were American geneticists at the Rockefeller Institute, eventually proved that something that Griffith had discovered was something that was called DNA, deoxyribonucleic acid. They published their findings February 1st, 1944. Unfortunately, the importance of their discovery wasn't appreciated yet, and Avery died before their work could be honored with a Nobel Prize. Nobel Prizes are never given after someone dies. We're going to see that as a going theme in this chapter. So what Oswald Avery and his colleagues did was isolate and purify what that transforming principle that Griffith discovered was. He performed 3 tests on this transforming principle. Qualitative tests showed that our DNA was present. Chemical tests showed the chemical makeup matched that of DNA. Enzyme tests showed only DNA degrading enzymes stopped the transformation. So it's kind of like they worked backwards to get the answer. Next, in 1952, 2 geneticists at Cold Spring Harbor Lab, Alfred Hershey and Martha Chase, provided further proof. They worked with a DNA virus, a bacteriophage, that infects bacteria. So this is a virus that infects bacteria. Bacteriophages that were produced within the bacteria growing in a radioactive culture medium will themselves be radioactive. They tagged viral DNA with radioactive phosphorus, and they tagged viral proteins with radioactive sulfur. Why did they do that, you might ask? DNA contains phosphorus and proteins does not. Proteins contain sulfur, which DNA does not. So those radioactive phosphorus and proteins were going to give us the answer in the end that we were looking for. What they found was that the bacteria that was infected with the radioactive sulfur and the bacteria that was infected with the radioactive phosphorus, when they separated the protein from the DNA using, yes, just a plain old kitchen blender, they separated the two of those by centrifuge, and they examined them. What they found was that the tagged DNA was inside the only thing that was inside the bacteria. The tagged proteins were not. 8.2, the structure of DNA. DNA structure is the same in all organisms. Believe it or not, the DNA that's inside of you is the same DNA that's inside of any living thing. You may have heard that a banana contains roughly 50% of the same identical DNA that we have, and that is true. All living human beings have 99 point roughly 8 to 99.9% of the exact same DNA. So it's a very tiny amount of DNA that makes us different from one another. DNA is composed of 4 types of nucleotides. Remember, back in chapter 3, we talked about the different types of nucleotides. We talked about different nucleic acids. The nucleotides are the building blocks of the DNA. So those 4 types are made up of the basic parts. The DNA is made up of a long chain of nucleotides. Remember, that's a polymer. The nucleotides would be the monomer. Each nucleotide has 3 parts to it. There's a phosphate group. There's a deoxyribose sugar. It's where DNA gets its name. It's the type of sugar that's used in it. And finally, a nitrogen-containing base. That's the part that makes the nucleotides different from one another. So this is just a basic structure of a nucleotide. There's nothing fancy here. We didn't put in all the little elements to confuse you. This is the basic structure, and it's the same one that's found in your textbook. The nitrogen-containing bases are the only differences in the 4 nucleotides of DNA. There are pyrimidines, which are single rings. You can see over here, there's thymine and cytosine. There's a single ring. We label that T, and the cytosine is C. So purines are double rings. So adenine and guanine. Adenine has 2 circles there. We label it with an A, guanine with a G. Watson and Crick determined that the 3-dimensional structure of DNA was what it was by building models. It took them quite a bit of experimentation and a little bit of stealing of some information from Rosalind Franklin. But eventually they realized that DNA is a double helix that is made up of a sugar phosphate backbone on the outside with bases on the inside. If you watch The Secret of Photo 51, you'll see that one of their original models they actually built it inside out. So those bases that are on the inside, like the rungs of a ladder, they had actually put those on the outside. Rosalind Franklin saw this and laughed at them. This is roughly 1951, 1952. They were building this model in their lab. Their discovery built on the work of Rosalind Franklin and Erwin Chargaff. Franklin took X-ray images using a process called X-ray crystallography. If you watch The Secret of Photo 51, it'll go a little more in depth with that, and you can see it. Those images suggested DNA was a double helix of even width. Chargaff's rules, so Erwin Chargaff up here, he worked in the same lab with Watson and Crick, and he noticed, because he was a mathematician, and you know, math people, they see patterns in numbers. He noticed that the amounts of adenine and thymine were equal to that of cytosine and guanine. So they must be paired together. Originally, they had thought that the purines paired together with purines and the pyrimidines with pyrimidines. But when they did that, they found that that DNA structure wasn't equal width when they did that. But if they paired an adenine and a thymine, so a pyrimidine and a purine, they would work. And so this is an image of that picture that Rosalind Franklin made. She was an outstanding scientist, but because of the time frame, it was 1950s, and we all know at that time, where were women supposed to be? At home, in the kitchen, married, not with a career. She had a brain that worked like a mathematician and a scientist, and so she was constantly searching for things. She made quite a few contributions to the field of science. This was probably one of her biggest. She took these pictures. She had 2 forms of them. There was a wet and a dry. And essentially you're looking at a cross-section of DNA. Okay, so they determined that this was a double helix. So it was a ladder that was twisted. Watson and Crick stole her idea, essentially, didn't give her credit, and they were published because they were men at the time and they knew the publisher. Their paper was actually published in the Nature journal way ahead of hers, all on the same publication date, but they were somewhere around page 300 and she was in the pages 700-800. So her paper wasn't read until way after theirs if you were reading front to back in this journal. What we know now is that nucleotides will always pair in the same way. That's very predictable. It's one of those laws of nature. The base pairing rule shows that nucleotides will always pair up in a specific way in DNA. We have adenine that pairs with thymine and cytosine paired with guanine. It always happens that way. No other way. Because a pyrimidine, which is a single ring, pairs with a purine, which is the double ring, the helix has a uniform width. The backbone, which is those sides to the ladder, is connected by covalent bonds. The bases are connected by hydrogen bonds. You can see that those bonds that hold it together here are hydrogen, whereas these are covalent. If you remember back to when we talked about biochemistry, you know that hydrogen bonds are very weak, while covalent bonds are fairly strong. DNA replication. DNA replication copies the genetic information of a cell. We remember when we talked about cell division that this is one of those stages of the cell cycle in which cells are going to make an exact copy of their DNA. A single strand of DNA serves as the template for the new strand. The rules of base pairing direct that replication. And DNA is replicated during that S phase. Remember that big chunk of our pie here. Each body cell will get a complete set of identical DNA. Proteins carry out the process of replication. DNA serves as a template for that, and enzymes and other proteins will actually do the work of replication. Essentially what happens is there's a molecule that will unzip the DNA. It's called a DNA helicase. It will unzip it, and then there are other molecules that will bring these little free-floating nucleotide guys in and attach them so that we end up with two identical strands of DNA. We call this semiconservative replication because we are sort of conserving the original strand. So half of that second strand is going to be part of the original. The DNA polymerase enzymes bond the nucleotides together and form this double helix. Polymerase enzymes form covalent bonds between nucleotides and the new strand. So there you can see a little bit clearer picture that polymerase, remember it's an ASE, so that means it's an enzyme. Polymer, meaning it's a complex made of parts, DNA. So it's a DNA polymerase. It's the enzyme that makes the DNA polymer. Its job is forming those new covalent bonds between the nucleotides, and then eventually hydrogen bonds will form between them so that you will have two identical strands of DNA. In this picture, you can see again, the original strand is the blue, the new strand is the orange. You can see how they are identical if you look at the colors, the reds, the greens, the blues, the purples. I will link this short video in the notes. It's a pretty good video. It's one of my favorite that we always show in class. And it will actually kind of show you a 3D replication of how DNA replicates itself.